Welcome to the deep dive. We take complex topics, cut through the noise, and really hand you the key insights. Today, we're plunging into something really fundamental, how our bodies think and move. It all comes down to electrical signals. We're talking about the basic electrical language of our cells. And our guide for this is Chapter 7 of Boron and Boulpaep's Medical Physiology, focusing on electrical excitability and action potentials. Our mission, really, is to give you a shortcut through this, well, pretty dense world of cellular electricity. We'll unpack the complex ideas, explain step by step how these systems actually work, and crucially, connect it all to real-world clinical stuff. So by the end, you should feel, confident, tackling even the tough parts. Ready to jump in? Okay, let's start at the beginning. What exactly is electrical excitability?
Right. Well, it's this really remarkable property. Certain cells have it, think neurons, your nerve cells, and also myocytes, your muscle cells. The key thing is when their cell membrane depolarizes, that means it gets less negative inside. And if it crosses a certain point, a threshold.
It triggers something big.
Exactly. An all or none response. That's the action potential.
All or none, like slicking a light switch. It's either on or off.
Precisely. There's no halfway. Once you hit that threshold, boom, you get the full response.
And this action potential, what does it look like? Is it just a blip?
Well, it's often called a spike. It's a spontaneous, really brief, regenerative electrical impulse. The voltage across the cell membrane shoots up incredibly fast, maybe 100 millivolts more positive than its usual resting state.
And that signal travels.
Oh, yes. It propagates over long distances. Think about information traveling from your fingertips to your brain, or commands going from your brain out to your muscles. That's action potentials doing the work. If we try to visualize it, imagine plotting the membrane voltage against time. You start negative, right? That's the resting potential, maybe minus 70 millivolts. When the action potential kicks off, you see this incredibly rapid, smooth rise, the depolarizing phase.
Shooting upwards.
Way up, yeah. Into positive territory, often peaking between, say, plus 10 and plus 40 millivolts. That bit above zero. We call that the overshoot.
Okay, so it goes up fast. What then?
Then it comes back down. That's the repolarizing phase. And it's usually a bit slower, returning towards the negative resting voltage. Sometimes it even dips slightly below the resting level for a moment. That's called an after hyperpolarization before it finally settles back down.
And the shape of this spike, is it always the same?
No. The precise shape, the threshold level, how high it goes, how long it lasts, that varies. It depends critically on a few things. One is the behavior of specific ion channels, these little molecular gates in the membrane, how they open and close, which depends on voltage and time. Also crucial are the concentrations of ions like sodium, potassium, calcium, chloride, both inside and outside the cell.
And the cell itself matters too, like its size or shape.
Yeah. Yes, the overall membrane property is its capacitance, resistance, the cell's geometry, they all play a part. And this shape variation is important clinically. Think about it. Nerve cells need very brief spikes for super fast signaling. Heart muscle cells, though, they have much longer action potentials. That's essential for driving those slow, rhythmic contractions. It's form-following function.
Okay, so that's the big, dramatic all or none signal. But you mentioned threshold. What if the stimulus isn't strong enough to reach it? Do we get nothing?
Not nothing, no. That's where graded responses come in.
Graded, meaning proportional.
Exactly. Unlike the action potential, graded responses are proportional to how strong the stimulus is. Small stimulus, small voltage change. Bigger stimulus, bigger change. At least up until you hit that action potential threshold.
And do these graded signals travel far?
No, that's a key difference. They tend to fade out pretty quickly as they spread from where they started. It's called electrotonic conduction. Think of like ripples spreading in a pond. They get weaker the further out they go.
Why do they fade?
Energy loss, basically. The signal leaks out across the membrane. Their time course is also different. It's exponential, governed by something called the time constant.
Can you give examples of these graded responses?
Sure. The response of a neuron to neurotransmitters at a synapse is often graded. Or how a sensory cell, like a photoreceptor in your eye, responds to light initially.
So the cell adds up these smaller graded signals until
or unless the total depolarization reaches that critical threshold. Once the membrane potential, passes that point, it kicks off this runaway process. Voltage-gated channels snap open, causing more depolarization, which opens more channels. It becomes self-reinforcing. That's why smaller sub-threshold changes just stay graded and fizzle out.
That makes sense. Okay, let's talk about firing again. If a cell fires an action potential, can it just fire another one immediately?
No, there's a crucial recovery time involved. This leads us to the concept of the refractory period. But first, it's worth noting triggering an action potential isn't just about how strong the stimulus is, but also how long it lasts.
Strength and duration both matter.
Right. A really strong stimulus might only need to be applied briefly. A weaker one might need to be held for longer. There's often an inverse relationship. The product of strength and duration is key, up to a point.
Okay, so back to this refractory period. What is it?
It's the time right after an action potential fires when it's either impossible or much harder to trigger a second one. There are two parts to it. First is the absolute refractory period.
Absolute meaning. Absolutely impossible.
Pretty much, yeah. From the moment the action potential starts until the membrane is almost fully repolarized, you cannot get another spike no matter how hard you stimulate it.
Why not? What's happening?
The main reason is that the voltage-gated sodium channels, the ones that cause the rapid upswing, are temporarily inactivated. They're shut down and unresponsive for a bit.
Okay, so that's absolute. What's the other part?
That's the relative refractory period. This follows the absolute period. Now, you can actually trigger another action potential,
There's always a but.
but you need a stronger stimulus than usual or one that lasts longer.
Why is it harder now?
It's largely because some of the potassium channels, the ones involved in repolarization, are still open. Their outflow of positive charge makes the membrane harder to depolarize back up to the threshold. You need a bigger kick to overcome that lingering potassium current.
This is all incredibly precise. How on earth did scientists first figure out the mechanics behind these signals? It seems impossibly small and fast.
Yeah, it's a fantastic story. The really groundbreaking work was done by Hodgkin and Huxley back in 1952. They won the Nobel Prize for it. They used a preparation that was, well, surprisingly convenient, the squid giant axon. It's unusually large, which made it possible to insert electrodes and measure things.
And what did they discover?
They laid out the fundamental ionic basis of the action potential. They showed it wasn't some mystical vital force, but rather voltage-dependent currents of specific ions, sodium and potassium, flowing through distinct molecular pathways or channels.
So ions moving across the membrane drive the voltage changes.
Exactly. At rest, the membrane's more permeable to potassium, which keeps the inside negative. During the action potential peak, though, there's this massive, brief increase in sodium permeability that flips the voltage positive close to sodium's equilibrium potential.
And then potassium brings it back down.
Right. Then an increase in potassium permeability helps restore the negative resting potential. It's this beautiful, coordinated sequence of opening and closing different ion channels.
But how could they tell it was sodium and potassium? How did they separate their effects?
Clever use of pharmacology. They found specific blockers. To isolate the sodium current, they used something called TEA, tetraethylammonium. It blocks the potassium channels.
So they could see just the sodium effect.
Precisely. And conversely, to see the potassium current alone, they used toxins like TTX tetrodotoxin, the famous one from pufferfish.
Right, the dangerous one.
Very dangerous, yes. Or STX saxitoxin from certain algae blooms, the red tides. These toxins specifically block the voltage-gated sodium channels. So, zap. No sodium current, and you can study the potassium current in isolation.
It's amazing how poisons became tools for discovery.
Isn't it? And what they saw was crucial. The sodium current, INA, turned on really fast, but then it also turned off quickly and inactivated. The potassium current, IK, on the other hand, activated more slowly with a noticeable lag, and then it stayed on. It didn't inactivate on that same fast timescale.
Which explains the different phases of the action potential.
Exactly. Rapid sodium influx for the fast upstroke, followed by sodium inactivation and the slower sustained potassium outflow for the repolarization. That sustained potassium current is why it's often called the delayed rectifier.
Okay, I think I'm following the overall currents, but you mentioned channels earlier. How did these broad currents relate to what individual channels are doing? These must be tiny.
They are tiny. But yeah, the macroscopic currents Hodgkin and Huxley measured are the sum of many, many individual channels opening and closing. Think of a single open channel. Its current flow is actually pretty simple, usually linear, like current through a resistor.
So if one channel is simple, how does the whole membrane give those complex current patterns?
The key is the probability that any given channel is open. That probability isn't fixed. It depends strongly on the membrane voltage. It typically follows an S-shaped or sigmoid curve.
Meaning, as the voltage changes, the likelihood of channels opening changes.
Right. So the total macroscopic current you measure is basically the number of channels available times the probability that each one is open times the current flowing through a single open channel. Their model was so good, it even predicted something called gating current, a tiny electrical signal caused by the movement of the channel's voltage sensor itself as it responds to voltage changes. This was experimentally confirmed later, which was amazing validation.
It's still incredible that their model, made before anyone had actually seen a channel protein, could explain things like threshold and the refractory periods.
It really is. They use these mathematical parameters N, M, and H to represent the probabilities of potassium channel activation, sodium channel activation M, and sodium channel inactivation H. By putting these together in equations, they could mathematically reconstruct the action potential shape with remarkable accuracy.
And how did those parameters explain threshold?
Threshold emerged naturally from the model. It's the voltage where the inward current through the opening sodium channels, driven by M, just starts to overcome the outward potassium current related to N and any passive leak currents. Below threshold, the outward currents win and the depolarization fizzles. Above threshold, the sodium current becomes self-reinforcing and you get the spike.
And the refractory periods.
Also explained by the parameters. The absolute refractory period is directly due to sodium channel inactivation, the H parameter dropping to near zero. Until H recovers, you can't get enough sodium channels to open for another spike. The relative refractory period happens while H is recovering, but the potassium conductance related to N is still elevated. That extra potassium outflow makes it harder to reach threshold, so you need a bigger stimulus.
This is fascinating. Let's zoom in now on the actual proteins, the voltage-gated channels themselves. There must be a huge variety.
Oh, absolutely. They're part of this enormous voltage-gated like ion channel superfamily. It includes the main voltage-gated sodium, calcium, and potassium channels, plus others. The journey to identify them involved using those toxins like TTX as molecular tags, purifying the proteins, and eventually groundbreaking gene cloning, like finding the potassium channel gene by studying mutant fruit flies called shakers.
Wow, shaker flies. Okay. And do these different channels look similar?
There's a common structural blueprint, yeah. Most have a core made of 6 segments that cross the membrane, labeled S1 through S6. The S4 segment is really key. It's packed with positively charged amino acids. It acts as the voltage sensor, physically moving when the membrane voltage changes.
And that movement opens the channel pore.
Exactly. The pore itself is formed mainly by the region between S5 and S6 called the P region or pore loop. This part is critical for determining which ions can pass through selectivity, and it's also where many toxins bind. And thanks to amazing advances in structural biology, like Roderick McKinnon's Nobel-winning work crystallizing a potassium channel, we now have detailed 3D pictures.
What do they look like?
Often like a pinwheel from above. A central pore made of 4 P regions surrounded by 4 voltage-sensing domains, the S1, S4 segments. There are linkers connecting these parts that couple the voltage sensing to the opening and closing of the pore gate. It's incredibly intricate molecular machinery.
Okay, let's dig into the specifics. Starting with sodium channels, what's their main gig?
Their primary job is driving that fast initial depolarization phase of action potentials. You find them in neurons, skeletal muscle, heart muscle, and they are extremely selective for sodium ions.
You mentioned ions earlier. Does anything else affect how they work?
Yes, interestingly, the concentration of calcium outside the cell has a significant effect on sodium channel gating.
How so?
High extracellular calcium makes the channels harder to open. It shifts their activation voltage to more positive values. This makes the cell less excitable. Conversely, low extracellular calcium makes them easier to open, shifting activation more negative. This leads to hyperexcitability.
And that has real clinical consequences.
Absolutely. Think about conditions like hypoparathyroidism, where you have low blood calcium. Patients can experience spontaneous muscle twitching or cramps tetany because their nerves and muscles are hyperexcitable. On the flip side, high calcium, maybe from hyperparathyroidism, can cause muscle weakness because cells are less excitable.
Are all sodium channels the same?
No, there's a lot of diversity. Humans have at least 10 different genes for the main alpha subunit. They're expressed in different tissues and have different properties, like sensitivity to TTX. For example, NaV1.4 is the main one in skeletal muscle. Mutations there can cause inherited muscle diseases like hyperkalemic periodic paralysis or certain types of myotonia.
What about the heart?
That's mainly NaV1.5. Mutations in that gene are linked to specific forms of long QT syndrome, which can cause life-threatening arrhythmias.
And in nerves.
NaV1.7 is really interesting. It's found in peripheral sensory neurons involved in pain. Mutations can either cause extreme pain sensitivity or, remarkably, a complete inability to feel pain. It really highlights its role in nociception.
We talked about toxins like TTX. Are there other important drugs that target sodium channels?
Yes, a huge class.
Yeah.
Local anesthetics. Things like lidocaine, procaine, tetracaine. These are workhorses in medicine and dentistry.
How do they work?
They reversibly block sodium channels, preventing action potential generation and propagation, especially in pain fibers. A key feature is their use dependence. They tend to bind more effectively to channels that are being opened frequently. So nerves that are firing repeatedly, like those carrying pain signals, get blocked more effectively than quiescent nerves. It's quite elegant.
Okay, super important channels. Now, what about calcium channels? How do they fit into the picture?
Calcium channels are also crucial, but often play slightly different, though sometimes overlapping, roles compared to sodium channels. Yes, they contribute to the depolarizing phase in some action potentials, particularly those with slower upstrokes or plateau phases. But they're also vital links in other processes.
Like what?
Big ones are excitation-contraction coupling, turning the electrical signal in a muscle cell into an actual contraction, and excitation-secretion coupling, linking depolarization in a nerve terminal or endocrine cell to the release of neurotransmitters or hormones.
So they let calcium into the cell, and that calcium acts as a trigger.
Exactly. Calcium influx through these channels acts as a critical second messenger signal inside the cell, initiating these downstream events. They are incredibly selective for calcium, even though there's way more sodium outside, and their gating is generally slower than sodium channels, which contributes to longer-lasting depolarizations, or the plateau phase you see in, say, cardiac action potentials.
You mentioned excitation-contraction coupling. Is that the same everywhere?
No. There's a really important difference between skeletal and cardiac muscle. In skeletal muscle, the voltage sensor is actually an L-type calcium channel, caveat 1.1, but its main role is not conducting much calcium. Instead, it physically interacts with calcium release channels inside the cell on the sarcoplasmic reticulum, so depolarization directly triggers internal calcium release. Extracellular calcium isn't required for contraction.
But art is different.
Yes. In cardiac muscle, depolarization opens a different L-type calcium channel, Cav 1.2. Calcium does flow into the cell through this channel, and it's this influx of calcium that then triggers the release of much larger amounts of calcium from the internal stores. This is called calcium-induced calcium release. So cardiac muscle contraction absolutely requires extracellular calcium.
Fascinating difference. And calcium channels also trigger release of neurotransmitters.
Right. In nerve terminals, depolarization opens voltage-gated calcium channels, often N, PQ, or R types. The resulting calcium influx triggers the fusion of synaptic vesicles with the membrane, releasing neurotransmitters into the synapse. Same basic idea in endocrine cells releasing hormones.
Are there different types of calcium channels like with sodium channels?
Yes, quite a few. Again, about 10 genes for the main subunit in humans. They're often classified by their electrical properties and sensitivity to blockers. We already mentioned L-type channels long-lasting. They activate at relatively high voltages and inactivate slowly. They're crucial for the cardiac plateau and muscle contraction coupling.
And these are the ones drugs target.
Precisely. Drugs like verapamil, diltiazem, and the dihydropyridines like nifedipine, amlodipine block L-type channels. They're hugely important for treating hypertension, angina, and some arrhythmias.
Okay, what other types?
There are T-type channels, transient. They activate at lower voltages and inactivate rapidly. They seem to be involved in rhythmic firing patterns in the heart and brain. And then you have the N, PQ, and R types. These are mostly found at nerve terminals, mediating that calcium entry for neurotransmitter release. They have specific blockers too, often toxins from snails or spiders, which have been invaluable research tools.
Are there diseases linked to calcium channel problems?
Definitely. Certain types of periodic paralysis involve mutations in skeletal muscle calcium channels. Lambert-Eaton syndrome is an autoimmune disease attacking presynaptic calcium channels, causing muscle weakness. And mutations in neuronal calcium channels are linked to conditions like familial hemiplegic migraine and certain types of ataxia.
All right. That leaves the third big family, potassium channels. You said they're the largest and most diverse and generally inhibitory. Why inhibitory?
Well, think about potassium's equilibrium potential. It's usually very negative, around N equals 80 or 90 millivolts, close to the typical resting potential. So opening potassium channels tends to drive the membrane potential towards this negative value, or keep it there. This counteracts depolarization caused by sodium or calcium influx.
So they stabilize the resting state and help end the action potential.
Exactly. They are absolutely crucial for repolarization, bringing the membrane back down after the spike. They help shape the action potential duration, control how frequently a neuron can fire, and stabilize the resting potential between spikes. They're the brakes, in a sense, opposing the excitatory actions of NAP+ and CA2+ channels.
Given their diversity, how are they classified?
It's complex, but historically they were grouped based on function. One major group are the delayed outward rectifiers, the classic KV channels, like the one in the Hodgkin-Huxley model. They activate with a delay after depolarization and allow potassium to flow out, hence outward rectifier.
Okay, the repolarizers. What else?
There are transient outward rectifiers, also called A-type channels. These activate rapidly upon depolarization, but also inactivate rapidly. They often activate at more negative potentials than the delayed rectifiers.
What's their role?
They play a big part in controlling the timing between action potentials, especially in neurons that fire repetitively. The amount of A-type current can influence how quickly the cell reaches threshold for the next spike, thus regulating the firing frequency.
Do these KV channels get blocked by drugs?
Yes. TEA, which we mentioned blocks potassium channels generally, is effective here. Another common blocker, especially for A-type channels, is 4-aminopyridine, 4-AP, and there are peptide toxins like dendrotoxins from mamba snakes that block specific KV channels.
And clinical relevance for KV channels?
Huge, especially in the heart. Mutations in genes for cardiac KV channels, like KCNQ1, KVLQT1, or CACNH2, H or RG, or associated proteins, are a major cause of inherited long QT syndromes. This leads to prolonged cardiac action potentials and a high risk of dangerous arrhythmias. They're also vital in the inner ear for hearing, so mutations can cause deafness.
Okay, what other kinds of K+ channels are there?
Another important family are the calcium-activated K+ channels, KCA. As the name suggests, their opening depends not just on voltage, but also on the concentration of calcium inside the cell.
So calcium coming in through calcium channels can then open these K+ channels.
Exactly. It's a feedback mechanism. Increased intracellular calcium makes these KCA channels more likely to open even at negative voltages. This leads to potassium outflow, hyperpolarization, which can help terminate bursts of action potentials or relax smooth muscle. There are different subtypes based on conductance, BK, big K, IK intermediate, and SK small k channels.
Interesting feedback loop. Any others?
Yes, the inward rectifier K+ channels. These are structurally simpler, often lacking the main voltage sensing domain. Their unique property is that they conduct potassium inward much better than outward, especially near the resting potential.
Why is that useful?
They are very important for setting and stabilizing the negative resting membrane potential close to potassium's equilibrium potential. They help clamp the voltage near rest and prevent excessive loss of potassium from the cell during repetitive activity.
How do they achieve that inward rectification?
It's largely due to blockage of the pore from the inside by intracellular magnesium ions and polyamines like spermine. When the membrane potential is positive inside, these positively charged blockers get driven into the pore, hindering outward K+ flow. When it's negative inside, they're pulled out, allowing inward K+ flow more easily.
Are these KIR channels regulated?
Yes, heavily. Some are directly gated by G proteins, GRRKs. For example, acetylcholine released from the vagus nerve activates GRRKs in the heart, slowing the heart rate. Others are regulated by intracellular ATP, the KK-ATP channels. These are inhibited by high ATP levels and open when ATP is low and ADP is high.
Linking metabolism to excitability.
Precisely. The classic example is in pancreatic beta cells. When glucose levels rise, ATP levels increase, closing KTP channels. This depolarizes the cell, opens voltage-gated calcium channels, calcium flows in, and triggers insulin secretion.
So drugs could target these?
Yes. Sulfonylurea drugs, used for type 2 diabetes, work by inhibiting pancreatic KTP channels, promoting insulin release. Conversely, drugs called K+ channel openers activate KTP channels and can be used to relax smooth muscle, for example.
It's an incredibly diverse and important family. Okay, we spent a lot of time on how the signals are generated within a cell or at a specific point. How do these action potentials actually travel down a long nerve axon or muscle fiber?
Right, propagation. It relies on something called local current loops. When one patch of membrane becomes active depolarized during an action potential, there's a temporary excess of positive charge just inside that region.
And that positive charge spreads.
It flows passively along the inside of the axon to the adjacent, still inactive resting regions. This local current flow then depolarizes that neighboring patch of membrane.
Enough to reach threshold.
Exactly. If the local current is sufficient to bring the adjacent membrane to threshold, it triggers a brand new, full-blown action potential right there. And then that active region generates local current to excite the next patch, and so on.
So the signal continuously regenerates itself down the line.
Yes, that's the key. Unlike those graded potentials that fizzle out, the action potential is actively regenerated at each point, allowing it to travel potentially meters without losing strength or amplitude.
That seems effective, but maybe not super fast. Has evolution found ways to speed this up? Nerves need to be quick.
Absolutely. The nervous system has evolved two main strategies to make propagation much faster and more efficient.
What's the first one?
Simply increase the axon's diameter. A fatter axon has lower internal resistance, meaning those local currents can spread further and faster along the inside. The classic example, again, is the squid giant axon. It's huge, up to 1 millimeter wide. That allows for pretty fast conduction, around 25 meters per second, even without the second strategy. Good for rapid escape reflexes.
But our axons aren't usually that big. What's the vertebrate solution?
The second, and arguably more sophisticated strategy, is myelination.
Ah, the myelin sheath, that fatty insulation.
Exactly. It's formed by specialized glial cells, Schwann cells in the peripheral nervous system, oligodendrocytes in the brain, and spinal cord wrapping layer upon layer of their own membrane around the axon, sometimes hundreds of layers.
How does that help?
Two main ways. First, it dramatically increases the electrical resistance across the axonal membrane, so less current leaks out. Second, it significantly decreases the membrane capacitance, meaning less charge needs to accumulate to change the voltage.
So the insulation keeps the current flowing along the axon.
Right, but the myelin isn't continuous. It has these regular gaps.
The nodes of Ranvier?
The nodes of Ranvier, yes. These are short, unmyelinated segments of the axon membrane spaced periodically along its length. And crucially, these nodes are jam-packed with voltage-gated sodium channels.
So the action potential only happens at the nodes.
Pretty much. The electrical current flows very rapidly and efficiently underneath the insulated myelinated segments from one node to the next. It's only at the nodes that the membrane depolarizes enough to reach threshold and regenerate the action potential.
So it jumps.
It effectively jumps from node to node. This is called saltatory conduction, from the Latin saltare, meaning to leap or jump.
And this is much faster.
Oh, much, much faster. In large, myelinated mammalian axons, conduction velocities can reach up to 130 meters per second. It's also more energy efficient because ions only cross the membrane significantly at the nodes.
There's a way to model this mathematically, right? Cable theory?
Yes, cable theory provides a framework for understanding how current flows along structures like axons, treating them sort of like leaky underwater telegraph cables.
What are the key concepts from that?
Two main parameters help us understand propagation speed. One is the length constant, usually symbolized by lambda. It tells you how far a subthreshold voltage change will spread electronically before it decays significantly, down to about 37% of its initial value.
So the longer length constant means the signal spreads farther.
Correct. And the length constant increases with axon radius and membrane resistance, but decreases with internal resistance.
What's the other parameter?
The membrane time constant, tauum. This reflects how quickly the membrane voltage can change in response to a current pulse. It's the product of membrane resistance and capacitance.
And faster is better for propagation.
Generally, yes. A shorter time constant means the membrane voltage changes more quickly, allowing the action potential to propagate faster.
So how does myelination fit into this? How does it improve speed?
Myelination is brilliant because it improves both parameters in ways that favor speed. By increasing membrane resistance, it increases the length constant, so the current spreads farther between nodes. And by drastically decreasing membrane capacitance, it significantly shortens the time constant, allowing the voltage at the nodes to change much more rapidly. The net effect, especially for axons bigger than about 1 micrometer in diameter, is a huge increase in conduction velocity compared to unmyelinated axons of the same size.
And the devastating clinical relevance of this comes into play with diseases that damage myelin.
Exactly. In demyelinating diseases like multiple sclerosis, MS, the immune system attacks and destroys the myelin sheath.
What does that do to the signal?
Losing myelin dramatically increases the membrane capacitance and decreases the resistance in those segments. The local currents generated at one node might then become too weak to depolarize the next node to threshold because too much current leaks out or is used charging the now bare membrane.
Does the signal just stops?
The action potential can fail to propagate. It gets blocked. This conduction failure is what underlies the wide array of neurological symptoms seen in MS problems with vision, movement, sensation, coordination, depending on which pathways are affected. It's a stark illustration of how critical myelin is for normal nervous system function.
Wow. We've certainly covered a lot of ground today, from the fundamental all-or-none nature of the action potential through the intricate workings of different ion channels, sodium, calcium, potassium, and finally to how these signals speed long myelinated nerves. It's really amazing to think about these tiny molecular gates, opening and closing in precise sequence, orchestrating everything from a simple reflex to our most complex thoughts.
It truly is. And maybe a final thought for you to consider. We've seen how incredibly elegant and finely tuned this whole system of electrical signaling is. Every sensation, every movement, every thought depends on this precise dance of ions and channels. So what does it imply for understanding disease when even a tiny disruption, a mutation in a single channel gene, or damage to the myelin insulation can have such profound and widespread consequences on how we function, on our health, on our very experience of the world?